Hello and welcome to the Veterinary Anatomy Brainry. And today we'll be discussing about the histology of endocrine glands. Endocrine system, as you know, that comprises of the glands in the tissues or the solitary cells that will lay their secretions directly into the blood. And the secretion is known as the hormones, has been derived from the Greek word known as to set in the motion. Now, this endocrine system, along with the autonomic nervous system, will work together, and will coordinate and control the metabolic activities and internal environment of the body. So these endocrine glands, I told you, these are the ductless glands. Rather than directly pouring their secretion into the blood, they will first secrete their secretion in the interstitial fluid. And from the interstitial fluid, the blood will take the secretion towards the specific organs. And some of the hormones have the single target, and some will have the wide spread effect. And the distribution of these endocrine glands in the body, you have the major endocrine glands in the body as thyroid, parathyroid, and pituitary gland, and some other glands also. You have the part of the organ that acts as endocrine gland. You have the part in pancreas as islet. You have the testes. There is a part which acts as endocrine glands. Ovary, there is a corpus luteum. In placenta will also act sometimes as endocrine system. In the kidney, there are some part that will act as endocrine organ. Besides these, you have the small solitary cells or the cells in some parts of the body which singly act as the endocrine system, or we call it the neuroendocrine system. You have the APUD, amine precursor uptake decarboxylase type of cells. And you have the examples there, the lining epithelium of GIT. There are some of the solitary cells, they will act as the endocrine system. In the respiratory tract also, there are some of the cells that will secrete the endocrine secretions. So hormones, based on their chemical structure, you have the four types in the body: the amino acid derivatives, the small peptides, proteins, and the steroids. Examples are given. You can see that adrenaline is amino acid, small peptide encephalin, thyroid is a small peptide, proteins, insulin is a protein in nature, is hormone. Then the progesterone, estrogen, they are the steroidal in nature. The development of the gland will take, you have this membrane, cell membrane, the developmental phase, and some of the cells will get the signal to form the cord-like structure. And in the right, you can find here there is a invagination of the cells, and this invagination of the cells basically forms a cord-like structure having the two ends. One is called as the proximal end, that is towards the membrane, and one is the distal end, that is away from the membrane. This proximal end basically ruptures here, and this gland will get separated from here. And the nature of the endocrine gland is that endocrine glands is basically is that they are richly supplied with the blood supply, blood vessels. So the moment the gland, this the distal part will get separated, it will be supplied by the blood vessels or the blood capillaries, and they will lay directly their secretions into the blood. So types of endocrine glands, basically on the basis of the how the cells are arranged, you have the cord and clump type, where the cells in the endocrine glands are arranged in the cords or the clumps. You have the one more type that is called the follicular type. The cells are arranged in the follicles. So the difference between the two regarding their secretion is that in the cord and clump type of these endocrine glands, the secretion will be laid directly into the blood, whereas in case of the follicular type, the secretion will be produced by the cells and they will store it first, then from the storage it will go towards the blood. That means it will go indirectly, the follicular type, and in the cord and clump type, it will go directly into the blood. You are the best example of cord and clump. The many endocrine glands, pituitary, adrenal, parathyroid, and the pineal glands, they are the cord and clump type. While the follicular type, you have the best example in the body as the thyroid gland. And what I told you, the secretion, you have the extracellular method, while in case of a cord and clump, you have the intracellular method type of the secretions. So these are the major endocrine glands in the body that we have already discussed. So we'll be coming first to the pituitary gland, also known as hypophysis. Hypophysis cerebri is the other name of the pituitary gland. And this gland is known as a master endocrine gland, and is situated into the area in the body of a siphonoid bone is there, that forms the base of the skull basically. And in the base of this siphonoid bone, on the interior aspect of the skull, there is one area that is called as sella turcica. And in this sella turcica, on the arrows, you can see that this is the sella turcica area will be present in this pituitary gland. And because of the development of the pituitary gland, it has two parts. One is called as the anterior pituitary gland, and one is called the posterior pituitary gland. Anterior pituitary gland is also known as adenohypophysis, well as in case the The posterior pituitary gland is also known as neurohypophysis. Both the two parts of the gland are embryologically, morphologically, and functionally totally different. So if you see the development of this pituitary gland, what I told you that from the two sources it will come. Even the germinal origin is ectoderm in the both the parts, but the sources will be two. One of the sources will be coming from the mouth side, what you call is the stomodium. In the stomodium, there is a one area that's called the Rathke's pouch, and there will be the invagination of the Rathke's pouch. A diverticulum will be formed, and from the diencephalon, one more diverticulum will be formed. And in the later stages of the development, they will just combine together to form the anterior pituitary and the posterior pituitary. So these are ectodermal in origin. Both are ectodermal in origin, and but the sources are two. One is from the Rathke's pouch or stomodium, and one is from the diencephalon. So anterior pituitary, also known as adenohypophysis, basically is divided into three parts: pars tuberalis. Pars tuberalis is towards the stalk end, and then pars distalis is the major part, and then pars intermedia. Basically, this is the part that lies between the anterior pituitary and the posterior pituitary. Then posterior pituitary also you have three parts. One is called as a median eminence, that is almost in the level of pars tuberalis. Then you have pituitary stalk, what you call as infundibular stalk, and the main part what you call as pars nervosa. So this is the microscopic view. When you stain the tissue under the H&E, you can get the structure like this under the low magnification. You can see the adenohypophysis and the neurohypophysis, and entire gland is covered by the connective tissue, and this connective tissue covering you call as a capsule. Adenohypophysis, if you talk about the pars distalis and the pars tuberalis, we will see first. So the parenchyma of this part, adenohypophysis, the 75% is made up of pars distalis and tuberalis basically. And the cells will be arranged in the form, you can see it in the form of the clumps or the cords here. Irregular cords, basically the cells are arranged in the form of the clumps. And these small pinkish or reddish, what you see here, basically these are the capillaries, because the endocrine gland, and endocrine gland is a feature that is highly supplied by the capillaries. You can see these. So it is highly supplied by the capillaries, and the capillaries here will be the fenestrated, or the things will here will be those white space. You can see these are the sinusoids. So fenestrated capillary basically is a capillary which have and the irregular or which have discontinuous endothelium, but the outer basement membrane will be permanent. So this is high magnification of the pars distalis and the pars tuberalis. You can see by the arrows, the cells are arranged in the form of the clumps, and they are separated by here capillaries. You can see here in this arrow, there is a capillary here, there is a capillary. So many capillaries will be there here. So now if you stain the tissue with a different type of stains, you have based on the affinities of H and E, the cells of this pars distalis or pars tuberalis have been divided into chromophobe and the chromophils. Chromo means the stain, philia means attraction, and phobia means fear. That means there will be the some the majority of the cells which have the attraction towards the stain, and they will get darkly stained, those you call as chromophils, and there will be the some of the cells which will get light stained, those you call as chromophobes. You see here in the arrow, like these cells, they are lightly stained, you call them as chromophobes. Now these bluish and the pinkish color cells, you call them as chromophils. So if you look out the morphology of the chromophil cell and the chromophile cells usually are larger in number. So these bluish and pinkish are the larger in number. They are abundant in present, and they will stain intensely. While in case of the chromophobe cells, they will be smaller. You can find here the arrow, the smaller cell. This one is smaller cell with few secretory granules in the cytoplasm, and they will stain very, very less faintly. So these are the example of the chromophil cells. You can see deeply stained cells. CAP is the capillaries. You can see the capillaries here. Now chromophil cells, the type of stain we use based on the affinities for basic and acidic dyes, they will be further classified as acidophils and the basophils. So acidophils basically stain with the acidic dyes. You have the eosin and the orangey as an acidic dye, and basophils basically stain with the basic dye. You have the hematoxylin and the PAS. So here the bluish color basically is the basophils, and the pinkish usually are acidophilic cells. Again, these are the basophils. You can see these are the acidophilics. This CAP is the capillaries. So acidophilic cells, acidic dyes, eosine and orangey, and they will be round in morphology with cytoplasmic granules. And the hormones that these acidic or acidophils will produce, one is the growth hormone and one is the prolactin. Basophils, they will get stained by the basophil dye, hematoxin in the past. And you have the cells, those are the polygonal, you can see the bluish polygonal shaped cells. The hormones that will secrete is TSH, FSH, and LH. You have the thyrotrophs, you have the gonadotrophs, and the corticotrophs. So altogether, the acidophils will constitute the 40%. Example is somatotrophs and mammotrophs. Basophil will constitute the 10%. You have example of the corticotrophs, gonadotrophs, and thyrotrophs. So this is the section of the pituitary gland as pars nervosa, pars intermedia, and the pars distalis. So again, A is acidophil, B is the basophil, C is the chromophobe, light stained cell, S is the smooth muscle you can see here. So this is A acidophil, B basophil, C as chromophobe, S as a smooth muscle. Again here as smooth muscle, here smooth muscle here. B is a basophil, A is the acidophil, C is the lightly stained cell here, chromophobe cell. Now, adenohypophysis pars intermedia, that is towards basically the neurohypophysis. It is poorly developed in case of humans or in case of animals, it's not that much developed. And the basic difference here, you can see that you will find a vesicular nature the of this part, and this vesicular part will resemble like that of a thyroid having the colloid filled vesicles, and the cells will be the cuboidal in origin, cuboidal in structure, and the hormone it will produce is the melanocyte stimulating hormone. So you can see the posterior, pars intermedia, and pars anterior. You can see here one and two. You can see like that looks like that of a colloid follicle. Otherwise, it is a pars intermedia part of the adenohypophysis. Again, in this H&E staining, you can see here the asterisk mark is pars intermedia or intermediate lobe. You can see it looks like that of a thyroid gland, but it is a part of the pituitary gland. This is posterior pituitary, this is anterior lobe. Again here, the colloid fluid vesicles, you can see. Neurohypophysis, basically it will stores the hormones that are synthesized in the hypothalamus. And there is a relationship with that of hypothalamus by this part of the pituitary gland. You call that as hypothalamo-hypophyseal tract. And this part is basically the neurohypophysis and contains mainly unmyelinated axons of neurosecretory cells. And because the axons will be in this part, the cell bodies of those axons will be supraoptic and paraventricular that are in the hypothalamus. That's why it has a hypothalamo-hypophyseal tract. So the cytons or the bodies or the nuclei will be in the hypothalamus. The axons will, and those axons will be unmyelinated axons will be present in the neurohypophysis. Besides these unmyelinated axons, it will also consisting of the pituocytes. So this is how they are oriented by the hypothalamo-hypophyseal tract. You have the cell bodies or you have the nuclei in the hypothalamus, and the axons will go towards the posterior pituitary gland, and the secretions will be stored sometimes in the end of the axon, and you call them as a hearing bodies. So unmyelinated axon under the microscope under H&E, what you are going to see here like this. Then the capillaries will be there. Then the pituocytes nuclei you can find. This is the posterior pituitary. This part is the anterior pituitary, and this part, the follicular vesicular part, is the intermediate part of the pituitary gland. So this is the slide of H&E stained. The hearing bodies you can see that axons under the microscope, then capillaries, then pituocytes you can see. This was about the pituitary gland. So next is the thyroid gland. The thyroid gland has been derived from the word thyros, meaning the shield. It will just shield the pharyngeal part, or what you call is the neck part on the anterior aspect. You can see here it just shields the larynx. And shield, like what I told you, that the special feature of this endocrine gland is that it will store, manufacture, and store the hormone, and then It's release it when it is needed. That is the one of the biggest difference, that it will store its secretion first, then it will release, and it is supplied, the richest blood flow will be to this organ. So here it is a diagrammatic representation of the thyroid gland having the two lobes as right lobe, left lobe, connected by this what you call as isthmus. The microscopic view of the thyroid gland under the 4x, you will see like this. There will be the outer covering, what you call as a capsule, and inner parenchyma will be made up of the follicles, and some stroma will be also there. So this is the microscopic feature. The follicles will look like this, and the follicular cells will just form these rounded circles, and they will be having interior on the inner aspect, they will have the secretions what you call as the colloid or the thyroglobin. So then there will be the big cells, either they will be present between the area of the two follicles, or they will be present along with the follicular cells. Those are called as parafollicular cells. So thyroid follicle basically is a structural and the functional unit of the thyroid gland, and usually it is spherical in shape. So two types of the cells I told you. One is the follicular cells that will have the gelatinous homogeneous material inside, what you call as a colloid, and this colloid basically is a glycoprotein you call as a thyroglobulin. Then thyroid follicle or the thyroid gland, basically the lining epithelium or the thyroid follicular cell epithelium depends upon the activity of the gland. It can be cuboidal, it can be squamous type, or it can be the columnar type. That depends upon the gland is active or inactive. So in highly active glands, you have the amount of colloid because colloid will be used. So the colloid is used, so then there will be decrease in the colloid, but the follicular cells will become tall, that it will become the columnar type. In the resting gland, if the gland is in the resting stage, the colloid will be more, but the follicular cells will become flat or the squamous type. So this is how these resting gland, highly active gland, and moderately active gland have the difference between the two. That depends upon squamous epithelium or cuboidal epithelium or the columnar epithelium. The colloid I told you, it is a thyroglobin. Thyroprotein is it is there, and it will be stored in these thyroid follicles. Then it will be taken by the what you call as iodine uptake, and it will be taken by the blood. So this is the example of the thyroid follicular type of the follicular type of the endocrine glands, where the secretion is first stored, then it is released into the blood. Thyroid stroma, basically the connective tissue and the other things that will just surround these follicles, you call that as thyroid stroma. Capillaries will be very high because this endocrine gland, so blood supply will be very high there. So this is the thyroid stroma. Then parafollicular cells. What I told you, the parafollicular cells will be either isolated between the two follicles, or they will be between the follicular cell and the basement membrane. So here are the examples of how they will be present in the thyroid gland, and they will secrete the hormone that is called as calcitonin, and that will inhibit the osteoclast activity. This is the example under the microscope. What you are going to see: thyroid follicle with epithelium, with the colloid, and loose connective tissue, what you call as the stroma. Again, it's a high magnification. You can see the C cells or the parafollicular cells between the two follicles. And the follicle with a lot of colloid, that means it is in inactive stage. So these are some of the changes, some of the things that will happen in the hypothyroidism, and this will happen into the hypothyroidism. You can just go to any book or refer to any. You can see the many more things there. Parathyroid. Para means near, thyroid means gland. That means thyroid gland. That means these parathyroid glands are near to the thyroid gland, and they basically are the small yellowish color bodies. Usually the number will vary from 2 to 4, 2 to 6 in number, but mostly they are 4 in number, and they will be arranged 2 on the superior aspect, 2 on the inferior aspect. So parathyroid chief two cells you can find, or main two cells you can find. One is called as a chief cell or principal cell, and one you call as the oxyphil cell. These are the two main cells that will be present in the parathyroid gland. So chief cell or the principal cell usually are the polygonal in shape, and they will be arranged in the form of the cords or clumps. You can see this the cord here. So here it will be a clump. So irregular cords or clumps, they will be arranged, and they will secrete the hormone, parathyroid hormone or the parath hormone. So oxyphil cell, they are the larger as compared to that of the chief cells. They are very fewer in number, and they will be arranged in the clumps. The function, you can refer to any physiology book to see the function of this. So this is the H&E stained. You can see the oxyphil cells, the capillaries. So this is how physiological activity is there, how they will act, the calcitonin and how the parathormone will act. These are some of the effects of the hyperparathyroidism. So this is some applied anatomy of when there is a hypoparathyroidism. Coming to the adrenal gland or the suprarenal gland, they are the supra. They are on the top of the top pole or anterior pole of the adrenal gland of the kidney, and usually the size will vary in the different animals, but usually 4 to 6 centimeter long in case of humans. And adrenal gland, basically histologically, they have the two components. Even if you get cut in the cross section, also you have the two components of the adrenal gland. One is the superficial cortex, and one is the deep, is the medulla. So volume of the cortex is always greater than that of the medulla. So this is how you see the adrenal gland under the thorax with the capsule, with the cortex, and with the medulla inside. So adrenal cortex, the cells will be arranged in the form of the cords, and there will be a lot of sinusoids to this adrenal cortex, and the cells of this adrenal cortex will be secreting the steroid secreting cells, having a lot of lipids, having a lot of lipid droplets, smooth endoplasmic reticulum, and the mitochondria. Adrenal cortex itself is divided into three zones. One is glomerulosa, one is fasciculata, one is reticularis. So this gland is also covered by externally by the capsule. Followed by the capsule, there will be the zona glomerulosa, then there will be the fasciculata, then there will be the reticularis, and after that there will be the innermost, that is the medulla. So this is the diagrammatic representation of these layers of the adrenal gland. So coming to the glomerulosa, so the cells will be arranged in the form of inverted U or the arc shaped. So in case of horse or in case of donkey, the cells in the glomerulosa are arranged in the form of arc. That's why this layer there is known as zona arcuata. So in case of horse, this is known as zona arcuata. In case of the rest of the animals, it is known as glomerulosa. And if you see here, it is the inverted U. Cell layer is inverted U. Here also it is inverted U. So inverted U-shaped cells will be there. That is a special feature of this layer of adrenal cortex. So this is how under the high magnification, you cannot see the complete U, but you can see the glomerulosa cells which have the basophilic cytoplasm, and they will deeply staining with the spherical nucleus. So basophilic cytoplasm and deep stained with having the cytoplasm. So zona fasciculata, the bigger portion of the adrenal cortex. So it constitutes about 65 to 80% of the cortex. The size, the cells are polyhedral in the nature, and the cells will be arranged in the form of the straight cords. They will be one or two cell thick. This cord is one cell thick. This cord and this cord has combined it together and formed the two cell thick cord. So this is like that, one cell thick or two cell thick cord, and these type of cells will secrete the glucocorticoids and the cortisol. Zona fasciculata. So zona fasciculata, the cells will be polygonal, single centrally placed. You can see single centrally placed nucleus. So many droplets will be, lipid droplets will be there, and you know that under the H&E, the lipid will not get stained. Because of that unstaining lipid droplets in the cytoplasm, it will get the whitish color of the cytoplasm. Here you can see here, and and you get the name of these cells as the spongocytes because of unstained lipid, and the cytoplasm will look like that of vacuolated, white vacuolated. You call them as the spongocytes. This is zona fasciculata. You see here the spongocytes white. Here also you can see zona reticularis. So this is the third part of the zona fasciculata. So it constitutes 10% of the cortex, and the cells will be polygonal in the nature. And the nature and the feature of these cells is that the cells are basically anastomosing in nature. That means here you can see under this rectangular part, you can see the cells are just combined one and another. They are just meeting one another, and they are just anastomosing with each other. And the cells will be smaller, and they will be darkly stained cells, and they will just secrete this steroid sex hormone, what we call as adrenal androgen. So this is zona reticularis. You can see the darkly stained cells, very darkly stained cells with anastomosis. Adrenal medulla, basically, that is a totally different, distinct function from that of the cortex. Has the two types of the cells. One is called as chromaffin cells, and has the two and one is called the ganglion cells. Chromophene cells will be the most numerous in this part, and the cells will be ovoid in nature. You can see the ovoid shaped cells, and they will secrete the catecholamines as acetylcholine and non-acetylcholine, or adrenaline or non-adrenaline, you call them. Why we call them as the chromophene cells? Because they will get stained with the chromium salt, and the reaction of the chromium salt, because of that, you have the chromophene reaction, hence it's called as chromophene cells. So this is adrenal medulla, how they will look. Sinusoid, again the capsule, the cortex, and the central medulla with this vein, high magnification under 40x. You can see that in the medulla cells here, the cortex cells. This will be the here, the reticularis part. Coming to one more gland, that is the pineal gland. So this is a religious ways of describing the genius. You have the seat of soul in the Western, in the Eastern philosophy. You have the eye of Shiva. So this has different names according to the religious perspective. So if you see the brain, the this pineal gland will be just present between the two cerebral hemispheres. So gross anatomy, it looks like that of a pyriform or the pear shaped, having the apex body stalk, and the maximum size it can reach to the animal or the human being is the 2 years. So this is about the pineal gland parenchyma. You will have the two types of the cells, the pinealocytes and the neuroglia cells. In the histological picture, you can see that. So this is the histological picture of the pineal gland as such. This is some blur, you cannot see anything here. Here you can see the pinealocytes or the chief cells, also known as. The shape of the pinealocyte, basically it is a star shaped cell. You can see the basically in the high magnification if you want to see. Basically you can see, and I think in the under electron microscopy you can see the shape of the cell. It looks like that of a star shaped cell, having the 4 to 6 processes. That's why it looks a star shaped cell. So one other thing you will find in the pineal gland, you call it the pineal sand, also known as brain sand, also known as you call it as corpora amnesia, and you call because there will be the deposition of calcium sometimes in the pineal gland, and because of that deposition of the calcium, you call that as the pineal sand. Coming to the pancreas, I told you in the pancreas there are the part of the pancreas that acts as endocrine part that you call as the islet, and you know the pancreas is a mixture exocrine and the endocrine gland, and these islets will act basically as a clumps. That why they are known as solitary or the isolated clumps. That's why they are known as the islets, and they have lightly as compared to that of the exocrine part. They will stain very less as compared to that of this is the low magnification. Under the high magnification, you can see this is the endocrine part, and this is the exocrine part of the pancreas. So islets basically in the body, you have three types of the islets: alpha cells, beta cells, and delta cells. Under an H and E, you can hardly visible see these three types of the cells because you have to go for the special staining for these type of the cells. But if you compare the percentage contributions, the beta cell will be more, having the 70% percentage in the islet as compared to the rest of the two cells. So this was about then some of the endocrine glands, the major endocrine, and some of the parts of the tissue that act as endocrine glands. And hopefully you will go through fully. You will go through this. And thank you.